For avian species living in polar and temperate zones, survival requires pushing their bodies to the absolute limits of their maximum metabolic capacity. They have to undergo massive physiological transformations to survive the annual solar cycle. But if the energy demands of these stages ever overlap, the math becomes fatal. The avian year is defined by three primary, highly energy-intensive states: reproduction, the complete molting of flight feathers, and long-distance trans-equatorial migration. To solve this energy bottleneck, species like the red knot and the ruff employ an extreme form of temporal segregation. This chart maps both species' life histories, revealing strict biological compartmentalization. Primary molt stops completely before pre-migratory fueling. Furthermore, we see hyperphagia reflected in sharp body mass spikes, scheduled exact weeks before migration. Birds schedule these changes based on an internal calendar, moving independently of immediate local weather patterns or sudden insect hatches. They prepare for environmental conditions that do not yet exist. The evolutionary viability of these species relies on an internal, anticipatory regulatory system capable of strictly isolating massive energetic demands months in advance. This presents a mechanical problem. A biological system needs a reliable method to anticipate seasons accurately enough to initiate structural physiological changes before the external environment actually shifts. Biologists identified the existence of an internal, endogenous biological clock by isolating African stonechats in a highly controlled laboratory environment. Researchers held these birds in constant equatorial conditions, 12 hours of light and 12 hours of dark, with no seasonal fluctuations in temperature or humidity, for over a decade. This graph tracks a single bird over 12 consecutive years in that static environment, continuing perfect cycles of gonadal growth and molt. Notice how the peaks of this free-running rhythm slowly drift leftward because the internal period is not exactly 365 days. Because this internal oscillator is slightly inaccurate, a bird in the wild requires a constant environmental syncing cue, a Zeitgeber, to prevent the physiological calendar from drifting out of phase with the actual seasons. The baseline rhythm is hardwired into the bird's physiology. The astronomical cue of daylength acts as the tuning mechanism, locking that internal clock to the solar year. It seems obvious that to measure daylength, an animal uses the photoreceptors in its eyes. In birds, this is incorrect. Decades of blinding experiments have demonstrated that enucleated and pinealectomized birds still exhibit perfect photoperiodic responses, accurately initiating gonadal recrudescence and molting on schedule. The true mechanism relies on the permeability of the skull. Ambient photons are capable of penetrating the bone and directly irradiating specialized tissue deep inside the avian brain. Here we see a microscopic view and a schematic of the specific sensory network responsible. These are cerebrospinal fluid contacting neurons that express neuropsin, or OPN5, a rhodopsin superfamily photopigment. Anatomically, this network is isolated in the mediobasal hypothalamus, or MBH. It operates completely independently of the visual cortex. The primary sensor for seasonal timekeeping is a dedicated photon counter located at the geographic center of the neuroendocrine system. We also assume that measuring daylength requires the brain to calculate the total ratio of light to dark accumulated over a 24-hour period. This diagram maps the framework for avian photoperiodic time measurement, the external coincidence model. Look at the bracket labeled photosensitive phase. This circadian window opens 11 to 12 hours after dawn. In the top panel, the short winter day ends before this window opens. In the bottom panel, the long summer day extends far enough to overlap it. Researchers proved this model by experimenting on Japanese quail. They exposed birds to extremely short days, keeping them in the dark for 18 hours. However, if they flashed an artificial 1-hour pulse of light precisely during that photosensitive phase, the birds exhibited full seasonal gonadal growth. Photoperiodic time measurement relies on internal synchronization rather than a simple tally of daylight hours. The seasonal biological cascade begins only if environmental photons hit the deep brain at the exact moment the endogenous photosensitive window is open. The timing of the light pulse, rather than the total volume of light, determines the physiological outcome. This schematic outlines the immediate trigger. Deep brain photoreceptors stimulate the pars tuberalis to release thyroid-stimulating hormone, or TSH, directly into the ependymal cells of the MBH. This TSH signaling throws a local genetic switch, upregulating the DIO2 gene and downregulating DIO3. The DIO2 gene encodes an enzyme that performs a highly specific function. It converts inactive thyroxine, T4, into biologically active triiodothyronine, T3. This creates a powerful, highly localized thyroid spike within the brain. The presence of active T3 drives a morphological change. It causes the physical retraction of glial end feet down in the median eminence. This neuroglial remodeling removes the physical barrier blocking gonadotropin-releasing hormone, or GnRH. With the barrier retracted, GnRH is free to flow directly into the hypothalamo-hypophyseal portal system. The abstract astronomical cue of extended daylight translates into a concrete structural modification of the brain's neuroendocrine barrier, providing GnRH direct access to the pituitary gland. Once the barrier drops, the endocrine cascade moves rapidly outward. GnRH reaches the pituitary gland, which in turn releases luteinizing hormone into the general bloodstream. Systemically, LH drives rapid gonadal recrudescence, the physical growth of the reproductive organs, and triggers the massive release of steroids, including testosterone and estrogens. These systemic steroids drive behavior and initiate seasonal neuroplasticity, physically reshaping the complex architecture of the bird's brain. Looking at these cross-sections of the Aucine song control system, we can observe the HVC and RA. During short winter days, shown on the left, these structures are heavily regressed. Following spring photostimulation, both undergo dramatic volumetric expansion. This physical expansion relies on two biophysical mechanisms, localized neuropil growth and de novo neurogenesis, where entirely new neurons are born and migrate directly into the HVC. The physical growth of these brain regions correlates directly with the complexity and frequency of the bird's vernal song patterns. The photoperiodic clock dictates the annual construction and demolition of the neural hardware required to physically produce the song. Systemic gonadal steroids do not account for all of this observed brain plasticity. Reviewing these bar charts on the right, we see data from castrated birds. Even entirely without gonads, these birds still exhibit significant photostimulated growth in both the HVC and RA. This points to parallel mechanisms operating independently of the gonads. Researchers have identified localized neurosteroid synthesis directly in the brain, alongside deep brain melatonin receptors regulating the song nuclei. Utilizing overlapping independent pathways provides an evolutionary advantage. It builds redundancy and fail-safes into the physiological execution of critical seasonal behaviors. The entire process forms an unbroken biophysical chain, beginning with the geometry of Earth's axial tilt and its orbit around the Sun. Photons pierce the skull, coinciding with an endogenous circadian phase. This triggers the DIO2 genetic switch, allowing for the release of GnRH and driving a systemic cascade that ends in de novo neurogenesis. Avian photoperiodism stands as a biologic logic gate, translating the mechanics of the solar system directly into the cellular architecture of an animal.